Please start. You are audible. Hello.
Yes. Good afternoon to everyone. Here I'm going to present about nutritional security through root vegetable crops. You know that what are the crops or comes under the root vegetable crops? Completely, the root vegetables such as carrots, radish, beetroot, turnips. These are the crops comes under the root vegetable crops. The primarily I'm going to cover carrots and radishes, how we are breeding for the nutrition security, as well as how we are enhancing nutrition through targeted breeding program. So before that I want to introduce that what are the root vegetable crops and what are the nutritions which are enriched with these naturally enriched with these all root vegetable crops, and what are the genetic resources are available in root vegetable crops, and how these genetic resources which will be used for breeding program, and how much amount of nutritions we can enhance through breeding programs by understanding of genetics through genetic resources, then how we can develop genotypes, and the genotypes how we can bring us a variety that will be useful for the the consumers. Because normally if you take these all root vegetable crops, particularly carrot and radish and beetroot and turnips, majorly this comes under the two groups. So that is Asiatic groups, another one is European types. So if you see primarily, if you see these carrots, particularly in the Hindu Kush mountains or particularly in the Caucasus mountains, especially in the Afghanistan region, there is the primary color of carrot color is black color. The black color which was taken by Roman empires from Afghanistan region to European regions. So original color of the carrot is black color, and these Roman people, they came to Afghanistan to conquer, explore the seeds which will be useful for fertility purpose. So that time the Apiaceae groups seeds like cumin and coriander and fenugreek, these seeds were collected from these Afghanistan regions. Along with that, the black carrots also they have taken to European country, European continents. So that once that carrots was taken to European regions, that is, originally the carrots was originated and evolved in the Afghanistan region. That original color of carrot is black color. The black carrot was enriched with anthocyanins. That anthocyanins means it is more than 500 compounds, that majorly cyanidin and cyanin groups which will be enriched with these black carrots, originally which are available in these Asiatic types. As well as these, because of these anthocyanins, so that whatever the carrots which are available in the Afghanistan region, those leaves which will be, the carrots which are available in the Afghanistan region, those regions, all carrots which have the, dark green leaves, as well as highly, so the segmented leaf is which will not be there. The leaves which are very, very closely observed in this Afghanistan carrots. But these carrots were taken to the European region, particularly which was introduced to the Netherlands regions. This original color of carrot, that black carrot, which got mutated into yellow carrots, as well as yellow carrots and orange carrots. That the farmers are royal family of Netherlands people who has selected these orange carrots, which was originally enriched with carotenoids. That original color of carrot is black color, which got mutated into yellow colors. That yellow colors means normally mustard yellow color, that is enriched with lutein content. Normally the lutein which will be used for macular degenerations, particularly eye-related diseases, that compounds which are used in the yellow carrots. But the yellow carrots which was not utilized by the European people, and from the yellow carrots that orange carrots was evolved. that orange carrots which are enriched with beta carotene as well as other carotenes like alpha carotene, beta carotene, zeta carotene, delta carotene, so that all type of total carotenoids enriched with these European carrots. So these European carrots, because of the European part which falls under the temperate region, these temperate regions means normally that whatever the carrots which are growing in this complete European region, the carrots are higher amount of water which will be there in this European carrots. Because of higher amount of water, that all nutrients, particularly carotenoids, which are diluted in this European carrots. So that European carrots are highly juicy. While eating European carrots, you will feel to eat the carrots. But when you eat Asiatic carrot, particularly the carrots originally is evolved from Afghanistan region, those carrots where the low water content will be there, because which are grown under the tropical region or Asiatic region. This Afghanistan, India, Pakistan, these all regions which falls under the tropical regions, or we can say that it is an Asian land. So these all carrots where water content is very low, as well as because of water content is very low, but inversely it is highly concentrated with anthocyanins. But in case of European carrots, the European carrots was highly enriched with beta carotene. Now you understand the two type of carrots, that is tropical carrots, another one is temperate carrots. So temperate carrots are temperate carrots has higher amount of water along with beta carotene. So once this orange carrot is evolved in the European region, these European people, they have distributed these orange carrots in the all over the world. So that the original carrots, which are if you see in the slides, the purple carrots are yellow carrots. The purple carrots was enriched with anthocyanins, and yellow carrots which are enriched with lutein content. That orange carrots which are enriched with carotenoids. Today, now we are talking about nutritional security, and we are talking about biofortification, and we are talking about all nutrients which we want to bring from wild species to cultivated species. So that originally whatever the carrots, if you go to is Afghanistan, or the traditional history of carrot history, as well as traditional history of these carrots, the originally that nowadays we are talking about, there is one new root vegetable crops that is called parsnip, Pastinaceae sativa. That root is which devoted of which does not have any beta carotenes. But the parsnip traditionally they consider as a carrot. But that after while evolution only they identified that parsnip is completely differ from carrots. So the carrots were if you go and see the civilization, as well as the geographical origin of these carrots, which particularly in these Caucasus mountain regions, you can find there is one species which is available, particularly in the carrots, that is queen anise lace. The queen anise lace carrot, which was the sparsely distributed in the Caucasus mountains, and that from that carrot only, which was evolved these black carrots as well as yellow carrots, then orange carrots was evolved. But due to the due to the higher amount of water, and due to the higher amount of beta carotene, the orange carrots were completely distributed throughout the world. Now, I was mentioning that traditionally that whatever the carrots which are there in the Afghanistan region, the leaves are dark green color. But when you go to European region, that carrots color has light green color, as well as the leaf appearance which will be there, highly dissected leaves which will be there. But not that much dissections will be there in case of tropical carrots, and temperate carrots which has higher highly dissected leaves which will be there in the orange carrots. So you can see in the slides that four colors which I am showing, that white carrots and orange carrots and yellow carrots and black carrots and red carrots. This red carrots also, along with white carrots, the red carrots also available in the Afghanistan region as well as western part of India. If you take India as well as Pakistan, these two countries, it is a secondary origin of carrots. That is, if you Haryana and Western Punjab, in these regions, naturally available the black carrots, which is used for the extraction of kanchi or the production of kanchi preparations, which are done commercially, and these carrots which are used for the animal feed purpose. But nowadays, due to the that after knowing about the that nutritional importance of these carrots, these black carrots, which was used for the extraction of anthocyanins, as well as which is used for the enrichment of anthocyanins into these normal carrots like orange carrots. That once these are the genetic resources which are available in all over the world. So only the four colors are very dominant. That is black carrot, red carrot, and yellow carrot, and orange carrots. These whatever the carrots which are evolved from this Asiatic region, this carrot is called the eastern carrots. But this eastern carrots means what? That Afghanistan carrots. But eastern carrots which went to European regions, that is Western countries, so that those carrots is called Western carrots. After evolving of this orange carrots, that orange carrot has the two groups. If you find in the European regions very small sized carrots, you can find. If you see here, you can see here, this is J and K. This is J and K. It is nothing but the small carrots. That is this group of carrot is called horn types. Horn types are the small short type of carrots which has the three groups. That is late season and early season and early short corns. So these are the carrots which are very, very popular in the Western countries. Even these Western carrots, particularly from the Netherlands region, that orange carrots which was taken to the America, North America, and North America, there is whatever the carrots which are grown in USA, those all carrots has longer roots. Those carrots are long orange carrots. The very the popular cultivars, of the baby carrots as well as long root carrots, which are cultivated commonly in the USA. So that you identified that Europe tropical carrots and tropical carrot to temperate carrots. The temperate carrots which has two types, one is long groups, another one is horn groups. Horn group means nothing but it is a small type of carrots, and long means that long root size will be there. That is more than 120 centimeter root length will be there in case of that long orange groups. So these all groups has only that orange colors, which in case of it is a root crops. If you take outer color and inner core color and outer core color, the center portion, it is a modification of root. So that the root portions means the xylem portions and phloem portions and the epiterm regions will be there. That epiterm regions and xylem portions and phloem portion with all colors are, there is no bifurcation in case of colors. You can see this in this slides that if you see in this the first groups, you can see here that yellow carrots, orange carrots, and red carrots, the root tips are very, very sharpened. But in case of temperate carrot, the root shape will be like blunt in nature. At the same time, when you cut the carrots, when you longitudinally if you cut the carrots, you can find that the clear color variations in the carrots. That is, you can see here that outer epidermis regions you can find the black, and center portions you can find yellow colors will be there. So this type of bifurcation, color dissections, which will be available only in the tropical carrots, but the type of variations which will not be there in case of temperate carrots. If you travel to Himachal hill regions or southern hills, like particularly Nilgiris hills, that whatever carrots which you can find, there is no color variations you can find. While eating carrots also you can identify. While eating the carrots, when you feel very hard portion in the center portion, that is called core, that is inner core. If suppose that inner core is very, very less portion is there, that carrots which will be comes under the European groups. But if suppose that portion is very, very hard and large size of inner core will be there as well as outer core will be there in case of tropical carrots. So these color variations as well as, there is another morphological variations you can find in these carrots. If you see in that outer portion of the carrots, means that the top shoulder, if you see here the shoulder region of carrots, which are the carrots comes under the tropical regions, those carrot has the typical green, green color shoulder will be there in case of black carrot as well as yellow carrots. So this type of the identical morphological features which will be there only in the tropical carrots, not in case of temperate carrots. So these color morphological variations you can find. So that this was I was explaining. The East Asian carrot, there is nothing but Afghanistan carrot which went to, there's Western regions. So the Western regions which was evolved Western European carrot, and it went to the England regions and Central region, particularly Russian regions also it went. And nowadays, nowadays that lot of breeding programs was done in the temperate carrots. The temperate carrots which was started cultivating in the North African regions also. So that comes under the North African carrots. So the carrots which are growing in India, those carrots are Indian carrots, or it is a black carrots and red carrots. If you come to North Indian plains, particularly Uttar Pradesh and Bihar, in these regions you can find only the black carrots and red carrots. This is the complete the illustration of horn groups and long orange groups. The horn groups, which is nothing but horn, early hot corn, early half long corn, late long half long corn, Paris market, Oxford, Nantes, Chandani, these popular cultivars, cultivated varieties which are very, very popular in the European regions. And long groups, if you take in the North America, I was mentioning that more than 120 centimeter length will be there. That carrots is very, very popular. That is imperator. That imperator which was evolved from the European carrots. That afterwards the rigorous breeding programs which has been done in the temperate carrots, and in Japan they have developed one tropical carrots, or we can say that carrots which can tolerate high temperature, heat tolerant types. That scientist who has developed, Dr. Kuroda, he developed Kuroda carrots. That carrots was taken to the South America, and they have developed root knot nematode resistant variety that is called Brasilia cultivars. So that Brazil they developed one cultivars, that is Brasilia, that is evolved from Japan origin carrot, that is heat tolerant types of Kuroda. The Kuroda which was rigorously intercrossed with wild types, which was brought into the root knot nematode resistant variety, that is Brasilia. Traditionally the South America, particularly in the Brazil regions, there is no cultivation of carrots, but because of this heat tolerant Kuroda types, as well as these Kuroda types was brought into the root knot nematode resistant cultivars. Even nowadays we have started cultivating the temperate cultivars and tropical cultivars in the that Australian regions also. So I was mentioning that how that morphological feature of tropical carrots and temperate carrots which will be there in terms of nutrition, and in case of breeding program what will be there. If you take carrot, radish, beetroot, turnip, these all are typically cool season crop. What do you mean by cool season crop? The cool season crops means only the sowing will be done at the end of the autumn season, but root production, particularly the root production of carrot and root production of radish and beetroot, turnip, these will be done only in the low temperature condition. It means that the economic part of these root vegetables which will form only in the cool season, cool temperature. That's why it's called cool season crops. These all cool season crops, all temperate crops which prefers when this it is basically botanically it is a biennial crop. This originally the carrots which was evolved from Afghanistan region, those carrots originally it is a annual in nature. Within a year which will produce the roots as well as flowers as well as seeds. But these carrots was taken into the temperate regions, and this morphologically which was adopted into this biennial nature. If you take radish also and beetroot also and turnip also, these also biennial growths. Biennial growth means that the first year which will produce the roots and second year which will produce the flowers. Means that these are all root crops. To enter into vegetative stage to reproductive stage, it's need very, very low temperature, or it's need to go for the vernalization or springization. Means that all complete roots of temperate roots which need to expose to 2 to 5 degrees Celsius around 2 to 5 weeks. So 2 to 5 weeks, these all roots when exposed to a very low temperature, that time only the roots which will produce the which will enter into the reproductive stage, that will produce the umbels. Then we can produce the seeds, or we can do the breeding program in case of carrot. I hope you understand till now that whatever the carrots, how this tropical carrots evolved into temperate carrots. So how the temperate carrots, temperate carrots need vernalization for the vegetative stage to the reproductive stage, but the tropical carrots does not need any type of vernalization. The annual types, so which can produce the seeds in the plains, but in case of temperate carrots, we can produce the seeds only in the temperate hills or only the highland regions only we can produce. So that IRI, we try to focus mainly the tropical based cultivars, particularly we developed that Pusa rutira, which has red color and higher source of lycopene, as well as the Pusa meghali, which is rich source of higher amount of beta carotene. In case of Pusa gulfi, that is white pale cultivar, which is a rich source of lutein content, and Pusa asita, which has the first the black color variety in India. We have introduced this variety, which is rich source of anthocyanins. That we have identified that cyanin compound and cyanidin compounds, these are higher amount of this will be there in the epitome. Even that why I am talking about these all 4 carrots. These 4 carrots, you can find that the epitome region, xylem regions, and phloem regions only uniform colors will be there. Only the core region, particularly in the xylem region, center portion of the regions, you can find that light white color discolorations will be there in the center portions. You can see I am marking here. So here you can find that the clear cut differentiations which will be there only in the tropical carrots, and also in these uniform epitome, xylem, as well as phloem colors. These 4 roots I have used for the breeding program. Why I used to? Because these see, nowadays we are talking about rainbow fruits, rainbow vegetables. So that it is not only colors. We are talking about edible colors. We don't want any synthetic dyes or synthetic food colors. We need natural food colors. These natural food colors we can extract from these type of vegetables or this type of crops. Normally, if red carrot is separate and orange carrot is separate and yellow carrot is separate and black carrot is separate. So in my mind, I thought that so we want to bring these all nutrients into the single carrot. How it is possible to we have this genetic resources, genotypes, or cultivars or variety which is released from IRI. So I utilized these 4 varieties and I started breeding. I started using these 4 cultivars as a parent. And I crossing red into orange and orange into yellow and yellow into black. So I have developed the populations like, I developed hybrids first. Then for example, red into orange, I developed F1, and orange into yellow, I developed F1, and yellow into black, and I developed F1. Then finally, all F1 I brought in the single genotypes, so that individual cultivars which has the outer color has some colors, and phloem color has another color, and xylem regions has some other colors. So these 3 regions, carrot is a modification of root. So that root portions, the 3 layers, 3 nutrients we can bring it. So we can talk about multicolored, multi-nutrition, and multivitamin and multi-mineral packed carrots in these crops. That individually I was talking about the white carrots which is enriched with lycopene. there is, and orange carrot which is enriched with beta carotene, and the purple outside the orange carrots. If I was trying to bring this type of types, outer color will be thickened, anthocyanins, and center core color will be orange colors, and red carrots is enriched in lycopene, and yellow colors which is used for the enriched with lutins, or particularly xanthophylls, which will be very, very high. And you can see here each nutrition which has different biological effects in the cancer, particularly the alpha carotene, which will be used for anti-proliferative, anti-metastatic, and beta carotene, which is used for the anti-inflammatory purpose, anthocyanins, and the polycyresinol, the polyphenols, which is used for the many which are very, very important role in the anti-cancer property. And we are talking about, see, I was talking about these all colors. So how this all colors which was evolved from individual, it is along, if you cross from Afghanistan to European regions, you can see the diverse colors. So first, now you understand the origin of the carrot, evolution of carrot, what are genetic resources are there, and how the genetics is working. That we have to understand. Once if you understand the genetics, then only we can go for breed the varieties. So that whenever we are using black carrot as a female parent or male parent, the black color which will express only in the epiderm region. Please keep it in mind that the purple color or black color are anthocyanins which will express only in the outer epiderm regions as a dominant. Whether we are using as a male parent and female parent, the black color will express only in the outer epiderm regions, as well as the xylem regions also, that anthocyanin will express. But in case of phloem regions, only that which parent lighter color we are using, that will express. For example, if suppose for example you are using black into yellow, that black will express in the epiderm region as well as xylem region. In phloem regions, which color will express? Only that yellow color will express. Instead of yellow color, you are using red carrots. If black into red, so the red will express in the phloem regions, not in the epiderm region as well as xylem regions. So, the same thing, when you are using orange carrot, that orange color will express only in the phloem regions, as not in the epiderm regions. So by these genetics, so by using of this crossing of these all four colors, we have identified that purple color is dominant over red, and purple is dominant over orange, and purple is dominant over yellow. So you keep it in mind that in case of carrot, only the darker color will express as a dominant, and lighter color will express as a recessive in nature. Because why you understand this color, this expression, that time only you can understand that what type of color is nothing but it is each nutrition. So that each nutrition we can bring in the layer by layer in case of carrots. So that normally that anthocyanins, which is this carrot, which can substitute for the orange carrots, as well as these anthocyanins product from the cardiovascular diseases, as well as which will product the cell death from unfavorable conditions. So these higher amount of anthocyanin we can enrich with purple orange and purple with white core and orange carrots. And these, normally I was mentioning that the purple carrots which has low water content but higher nutritions. But in case of as well as if you take in case of sweetness, the sweetness nothing but TSS, TSS which are very, very low in case of black carrot, which is very, very high, which is very, very high in case of tropical carrot, but which is very, very low in case of temperate carrots. But when you eat the carrot of temperate carrots, you feel to eat the carrots. But in case of tropical carrots, you don't feel to eat the carrots because of the reducing sugar and non-reducing sugars, which are very, very high in case of tropical carrots, but which are hidden by or which are hidden by these anthocyanins. So when these anthocyanins will be very, very high, you won't feel that even though it has high total soluble solids, that you cannot feel to eat these carrots. And you can see here these anthocyanins which has the ability to decrease the glucose levels. So that's why we can use for these black carrots also. That I was mentioning. Now, whenever we talked about black carrots, or you see here this black combination with yellow and black combination with orange and black combination of red. So this different combination is there. Only outer color is expressing as the black anthocyanins, and the inner phloem regions will be expressed only the lighter colors. So when we talked about these black carrots, so black carrots means that there is cyanidin compounds as well as cyanin compounds which will be there. But we have to talk always about availability of these anthocyanins. You can see here that by cyanidin 3 silosyl glucosides, first compounds, CY3XG, that is cyanidin 3XGG, that is cyanidin 3 silosyl glucosides. These all are readily, which can absorb in our body readily. Okay, but in case of other three, cyanidin compounds, cyanopyl glucosides or pyrolysyl glucoside, which can absorb only little bit. But the coumarin compounds which will not absorb in our bodies, but other compounds which are there. I was mentioning that anthocyanins means not one. Anthocyanins nothing but it's not one color. It is more than 500 compounds. These all five, and out of these 500 compounds, only majorly available that cyanidin and cyanin compounds. So you can see here that all colors, that expression, that anthocyanins which are masked by all carotenoids. So these are the purple is expressing in the outer color, but in case of lighter color, which is expressing in the inner colors. So these are the different cyanidin compound. This is cyanidin pathway. So how this bioavailability of each cyanidin groups which will be there in this. So what type of breeding program which we can use for the carrot crop? The carrot is highly cross-pollinated. So because of the cross-pollinations, we can get a lot of diversity in these crops. But when you go for the selfing, forcefully, if you go for the inbreeding of this, inbreeding of these crops, you can get you won't get 99.99%. You won't get that two type of the carrots. So we have to go for that what are the breeding program which will be applicable for the cross-pollinated crops, that is like mass selection, family selections, that we will apply. That when you see here, today when we talk about the carrots, I was mentioning that when we are talking about coreless character, coreless cultivar. When you bite the carrots, you have to find that hardiness should not be there in the center portion of the carrot. So we have to develop the coreless cultivars, as well as we have to develop the that if you find these carrots, that center portions, I was mentioning that tropical carrot has which has the typical feature of the chlorophyll productions in the shoulder portions as well as inner portions. So how we have to we have to breed the varieties. The chlorophyll should not come inside the core regions, and that what type of selections we have to go for this carrot breeding program. I know normally if you go for mass selection or family selection, that compared to mass selection, the family selections which we can go for fast progress. But in case of mass selection is easier, but in case of family selection is more difficult. But if I was mentioning these all colors which are expressing, which are inherited in the single genes, simple inheritance traits, so we can go for mass selections. But that normally the family selections, which we have to maintain separate families, but mass selections they will not go for the record keeping. That you can compare. These are the varieties which are highly diverse with the colors as well as nutritions. Pusa kulfi is enriched with lutein, completely white pale colors, and Pusa meghali, which is enriched with beta carotene, and Rudra, which is enriched with lycopene, Vasuda and Vristi and Asita. That Vristi and Vasuda also is evolved from these other red colors as well as red color varieties. See, I was mentioning that I was using the 4 colors as a parent, and I was breeding this, that A into B, like black into yellow, and yellow into orange, and yellow into red. So that intercross way, I am developing the F1, and I'm joining the F1 into in single genotypes. Means by using of these techniques, so we have tried, we have developed some magic populations. You can see here. Whenever you find that when you are crossing into black into yellow, you will get F1 as the purple is dominant and inside will be yellow color. But when you do the selfing of F1, the F2 populations, you will get a lot of colors from the dark black color to lighter colors. So dark black color means, the dark anthocyanins means like dark purple and light purple. These 2 type of colors will be there as well as in yellows, not only 2 colors, but four group of colors will come from the F2 population, and 2 group of colors which will come from the F2 populations. So by this process, we can fabricate and we can see that very attractive purple colors carrot, as well as medium purple carrots and high purple carrots. By looking of attractive way as well as nutrition way, you can look up the carrots and you can select the carrots for the consumer purposes. You can see here that the wider colors, you can see here in the plates that whatever carrots are which are there, that mostly that outer color will be black color and yellow. The center portions will be like yellow. So by these breeding programs, and we have developed a lot of genotypes which are enriched with black with means anthocyanin is enriched with protein content, and anthocyanin enriched with lycopene, anthocyanin enriched with this carotenoids. You can see here the different dark purple color and medium purple color, and even that upper portions dark purple will be there and lower portions light purple will be there, this type of lines. You can see here that orange also different color variations you can find. And you will see here that mass population, this magic populations, you can find the different colors of carrots, even these different colors of the carrot which has very sweety. That I was what we are looking that all black carrots which has the ability to mask the all sweetness. But whatever the combination of black into red and black combination of orange and yellow colors, this is giving very high sweetness, because the black color which is expressing only the epiderm regions. Because of this reason, that color this carrot has the highest sweetness, than the European carrot. It is from my experiment, so that I have identified that these are the carrot which has high sweetness. You can see here that I was mentioning that the high color variations you can find in these pictures. You can see here that dark colors, and this is top portions will be dark purple and lower portions will be light red colors, and here will be light purple color, and this is red color. These are parental types, but here the intermediate colors will be there. This we can go for the we can select for the breeding, we can select for the releasing as variety. You can see here the very attractive colors which has very good sweetness also, and these are the carrots which we can use for the preparation of pigment powder, and that powder we can use for the different as food additives and food colors also we can use. And these are the different combinations of some promising lines which we developed from the carrots, like Indian pollen carrot, a different series. And you can see these are the elite breeding lines which was developed by this breeding program. These are the now new emerging diverse carrot breeding materials, like attractive orange colors you can find, and medium orange colors with yellow colors. You can see here the different type of yellow colors. These all lines which are enriched with carotenoids, luteins and lycopene, carotenes, even that all vitamins and all minerals which are enriched with this one. As I was doing some experiments, that we want to map the anthocyanins, so that I have used around 440 primers for breeding programs. So out of 440 primers, the 7 primers which is showing the polymorphism for these colors, particularly anthocyanins colors, and which was expressing and which was expressing in the F2 populations, and we have mapped that P1 genes which was expressed, which was closely linked markers. We have different markers. Only these 7 markers showing the polymorphism in the F2 population, as well as that BSSR 43 and GSSR 137. These 2 markers are very closely linked with its anthocyanins. As well as these markers, we have replaced in the other populations also to identify the anthocyanin enriched lines. You can see here that whatever I explained, the black in the yellow, this type of colors you can see visually, so that you can easily understand. You can see here this is yellow into red, that F1 which has outer color will be red and inner color will be yellow. The F1 expression will be like this. But when you do the selfing of this one, you can see the diverse colors of carrots in the F2 populations. The similar way, when you cross with yellow into orange, then the F1 will be like outer color will be orange and inner color will be yellow. and F2 populations, you can find the 4 groups of orange and 2 groups of yellow. When you do the, this lycopene with black carrots, and outer color will be black and inner will be red. You can see here F1. When you do crossing of F1 again, and F2 will be the 4 groups of black and 2 groups of red, like 2 groups of light red and dark red, and dark black, also dark black, medium black, and even light pink colors we can find it. The similar way, the red into orange, that orange will express in the phloem regions, and outer will be only red will express. In F2 populations, you can find that outer color, the 4 group of red and 2 groups of orange, attractive orange is expressed. The similar way, we have developed a lot of populations. You can see here the Pusa Gulfi into Pusa Sida population, Pusa Sita into populations, and Pusa Gulfi into Rutira population, Pusa Rutira into Gulfi populations. These are the different colors of carrot, and these are the some promising lines which is derived from magic populations. And these are the different nutritional profiling of different individual lines. You can see here the lycopene expressions are very, very high in case of red carrots, as well as when you do the red carrots, when you do cross with black carrots also, which was expressing the higher amount of lycopene. And when you do the carotenoids, like orange color carrots, you can take the carotenoid rich carrot along with anthocyanins. These are the different mean performance of black carrots, as well as the hybrid combination for the horticultural traits as well as the nutritional traits. These are the, normally if you find the carrot, they was mentioning that when the tropical carrots or temperate carrots, the temperate carrots, when you enter into the when enough fertilization is there, it will enter into the reproductive stage. So what type of flowers which will come from this carrot? It is typically that umbel, compound umbel type of flowers will come. That normally the carrot umbel which will express only, what type of flowers which will be there in the carrot, only that anthromonoecious form will be there. Which we once after 90 days after harvesting of the carrot, what we will do normally, if you take radish or carrot or beetroot or turnip, that radish means 70 to 90 days we will harvest, and the carrot means we will harvest after 90 days to 120 days. In case of beetroot also similar way. But once you harvest, you will go for the selling purpose. But as a breeding point of view or a seed production point of view, once you harvest the roots, and based on the morphological traits, based on the economic traits, you will select the roots and you will make stecklings. Steckling means only top portion of the roots you will keep and bottom portions you will leave, as well as along with leaf stalks you have to keep, and you will plant stecklings into the field. That once the stecklings, when you will plant the stecklings, after harvesting immediately at the end of the January, you will plant the stecklings, and the stecklings will get started bolting. Bolting means once that roots will get bolted, flower will come. That compound umbel, primary umbel, and secondary umbel, and tertiary umbel. That primary umbel, secondary umbel, which will give very good seeds, the quality seeds. And these 2 umbels only which we have to use for the hybridization purpose as well as breeding purpose. Even that 4th and 5th cycle, whatever the umbels are coming, which will not produce any quality seeds. So that the primary umbel and the secondary umbels, what type of flowers will be there? Bisexual flower will be there. Bisexual flower means that each flower which has androecium as well as gynoecium, but only androecium will mature first before maturation of gynoecium. So protandry type of flowers you can find in the carrot flowers. The same way that later stage, whatever that third cycle as well as 4th cycle, whatever the flowers are coming, primarily that anthromonoecious flowers, mostly the bisexual flower will not be there, only some few flowers, bisexual flowers, and most of the flowers will be only that androecium, only that male parts will be there and female parts which will not be there. So this type of flowers express will be there in case of carrot, as well as if you see that that one type of male sterility or cytoplasmic male sterility which are observed in the carrot, that in that also there is many type of male sterility is there. Out of that petalite type of male sterility which is used for the hybridization purpose. So here, by using of CMS line, and we have developed some of the CMS based hybrids also here. See now, still now, how that how we have developed the populations, how we are selecting the lions along with enriched with anthocyanins and other nutritions. In the similar way, radish also has two groups. That if you say that Indian point of view, that we are talking about a rat tail radish. Normally, the rat tail radish, which will not produce any type of roots, the parts which will be used as the economic parts, or which will be used as the edible part. But when we go to the China or Japan, you can find that that large shaped radish, or it is nothing but daikon radish. Daikon, daikon radish. The daikon radish, which look like watermelon radish. Watermelon radish means the top portion will be green and lower portions will be white. But when you cut the radish, and inner will be like pink color or purple color will be there. That those type of radish which are available in Japanese regions, particularly in the Sonikama Island. When you travel to Sonikama or Japanese coast. In Japanese coast, you can find the Japanese radish that is called Raphanistum type of radish. But when you go to the European side, where particularly in the Mediterranean Sea, you can find the temperate type of radish. The temperate type of radish which has a round-shaped radish. Normally what we will see in the South India, we will see that large root radish which has a dirt white color. The dirt white radish which has high pungency. But when you come to North India, or when you go to the hill station, you can find the milky white radish, that isothiocyanate, that is pungent principles which are very, very less in case of temperate regions or subtropical regions, but which will express only in the tropical regions. So the tropical radish has the potential of pungent capit, as well as the tropical radish which has the dirt white color, as well as the root will not be blended, and it is the tapering type of roots will be there. But when subtropical regions as well as temperate regions, whatever the radish are there, those radishes are bicolored, round shaped, as well as blend shaped, and those radishes has high water content, and those radishes which has higher amount of vitamin C. So I was utilizing this. I was working in CITH from 2015 to 2019. I went to almost all districts. I collected some of the genotypes, particularly in the Dal Lake. Also, I have collected some of the radishes which will be utilized for the breeding program, because these small radishes, like round type of radishes, or we can say that it is a table radish. In South India, in our South India, normally the radish we can't eat because of the higher pungent, as well as the radish which has that unpleasant smell which will be there in this tropical radish. So when you eat radish from the Jammu Kashmir Dal Lake radish, or hilly regions, Himachal Pradesh or Darjeeling radish, these radishes are very sweet, as well as these radishes does not have any pungent principle. So I thought of targeting these types, like non-pungent radish, into the tropical types. So I started using the pink and purple and white, and top portions will be green and white. So I started crossing like radish, carrot only. I started crossing this one, and I got the typical typical colors in the outer portion as well as inner portions. Normally, today we are talking about nutritional security. So it is my point of view in terms of we already targeted the yield, but we have targeting the targeted nutritional breeding. We have target. Why we have to use because in terms of yield, we have to talk about per hectare this much kg of androgenes, this much kg of lycopene. So what if you go to pharmaceuticals, if you go to the nutraceuticals industry, they are expecting that any natural sources, natural plant sources, which will be there for extracting beta carotene or lycopene tablets or anthocyanin tablets, anthocyanin colors. So these colors we can extract from the radish and carrot, so that that flesh will be has anthocyanins more. So for that purpose, that that we have IRI, we have released Pusa gulafi and Pusa gulafi radish, that is purple pink flushed radish. So I used that radish as a parent, and I brought purple flushed, and you can see here purple flushed and pink flushed radish. You can find these are highly enriched with anthocyanins as well as vitamin C. You can see these are the lines which has the higher amount of anthocyanins as well as vitamin C. Normally the green radishes which has taste wise very good. You can see here these are the the very large size roots. Even per root size is around 5 kg or 10 kg of size is there. And this the type of radishes which has higher amount of weight, as well as this type of radishes which we can recommend for the industry purpose. We can extract the starch, we can extract the sugar from these radishes, and which will be utilized for the diabetes and other other pharmaceutical and nutraceutical industries. You can see here these are the lines which are enriched with anthocyanins and purple fleshed and pink fleshed radish. Okay. And see my point of view is that we by using of first we have to get the genetic resources. Once you get the genetic resources, we have to understand the genetics. Once if you understand the genetics, then we can go for breeding program or breeding strategies. So that by breeding strategy we can develop the breeding population. Once you develop the population, then that we can use it for the by using of markers, by using of metabolites profiling, or by using the RNA profiling we can do in these populations, or DNA profiling we can do in these populations. Then we can go for characterization of these all anthocyanins and lycopenes and these all minerals, so that these targeted nutritional breeding which will be used for the nutritional security. So that we have to target for the future breeding program. So we have to bring the nutritional enriched or nutritionally available wild types, that we have to bring into the cultivated types, so that it will be used in the cultivated varieties. So that we have to develop as a variety, then we have to give to industry, or we have to give to the farmers, or we have to give to the consumers. So everybody is getting the high quality, everybody is getting large sized radishes and carrots and beetroot and turnips, but we have to look up for the nutritional aspects. That was my ultimate aim. Thank you.
Thank you, Dr. Silva Kumar, for sharing such a valuable insights with us today, sir. I'm Shoaib here. Dr. Shoaib.
Oh, hi.
Sir, are you fine?
Yeah, fine dear. How are you?
Thank you, sir.
And sir, it was a wonderful lecture, sir. Thank you for taking time to.
Participants, if you have any questions, please ask.